Good afternoon, everyone. It's my great pleasure to introduce Shilpa Singh. Shilpa is a PhD student in my program. She did her undergrad in Haryana, India, and did a master's in weed science at the University of Arkansas in Fayetteville with Dr. Nilda Burgos. After that, she did a research assistant position in my program prior to joining her PhD in 2018 summer sometime. Shilpa has been a very active student in the bead judging contest preparation. She also helped with coaching bead identification for student preparations. And she also received the Outstanding Graduate Student Award from the department under the Agronomy category. Today, she's going to talk about her research on utilizing phosphite as a dual purpose fertilization as well as a weed suppression system in cotton. Shilpa, it's all yours.
Hey, Muthu, before you give it up, go back to that Teams group and tell them where we're really at.
Yes, I'll do that. Thanks very much.
All right. I hope you all can hear me.
Yes, we can hear you.
Okay. Good afternoon, everyone. Thank you, Dr. Muthu, for an introduction. And my PhD topic today to discuss with you all is phosphite fertilization for weed suppression in PtxD cotton system. So just to give a brief outline to the flow of this presentation, I'll start with a brief introduction, and then I will move on to the objectives. I will discuss the objectives and their methodology and results together. We'll summarize, and then finally the conclusions. Just to introduce you, we all know that weeds, they compete with the crops and result in heavy yield losses. To control weeds, growers heavily rely on herbicides. And the result of that is that we have the evolution of herbicide-resistant weeds. And now we have herbicide-resistant weeds, which is a serious problem in all of our cropping systems, and especially the cotton. As of today, we have 266 weed species which have evolved resistance to herbicides. And just to know that we have 31 modes of action of herbicides which are out and available in the market. And out of those 31, 21 are already weeds have evolved resistance to. So we need more effective and sustainable methods for the weed control. Now, the approach to weed control has been changing from just the herbicides to the involvement of the integrated weed management tactics and the crop rotation and herbicide modes of action mixing. And also with that, we also have the different traits of herbicide-resistant crops. On the similar lines, Dr. Kirti Rathod has a transgenic variety of cotton, which has this bacterial phosphite dehydrogenase gene that can convert the phosphite into orthophosphate, whereas weeds or in plants in general, which does not have this gene, they lack the ability to metabolize the phosphite, and hence they can be negatively impacted. Now you must be wondering that this phosphite and orthophosphate, how this is relevant. So we know that phosphorus is an essential macronutrient, and it is required for normal growth and development of the plants. And the plants, they can only metabolize the phosphorus in its orthophosphate form, but they are unable to utilize the phosphite form of the phosphorus. So therefore, when we adopt this phosphite, PtxD and phosphite technology, we solve the two purposes. It's kind of dual solution. One is the nutrient for the crop, and second, it's the suppression for the weeds. And that's what we want, something which benefits us more than the weeds. Based on the literature, phosphite is not new. It was there. It was used in different aspects. It was used as a fertilizer, as a biostimulant. Benefits of phosphite are termed as it reduces the phosphorus depletion. Mainly, it is used as a pathogen management tool in turf and citrus orchards, weed management, reduction of eutrophication, and it also is believed to reduce the evolution of tolerant weeds, which we would like to see, reduction in P pollution. So among all these, my major focus today is on weed management. And later on, when this technology is established, we would see if it will help with this aspect of reduction in tolerance to weeds. To achieve these idea of weed management, we had these 5 objectives established. The first one was to standardize the dose of phosphite as a pre-emergence application in soils with different levels of phosphorus. Determine the optimum dose of phosphite as a foliar application. Third, improve the efficacy of glufosinate in tank mix combinations with phosphite. Fourth, to evaluate the response of different crops and weed species to pre and post application of phosphite. Fifth one, to investigate the interactions between cereal rye cover crop and phosphite. So we tried to cover all the aspects where we can use phosphite and can get a benefit out of it. But for today's presentation, I will show the results from the first 3 objectives only. All these objectives were conducted under greenhouse environment and also in the field environment. We wanted to observe if there would be any variation in the results when we use this in the phosphite in the greenhouse, controlled environment, and also in the field when there is all kind of environmental factors play their role, and we will see how the phosphite will show its effect on weed management. But for today's presentation, I am going to show the results just for the greenhouse, which is already would be a lot to digest for you all. To begin with the first objective, phosphite as a pre-application. We had 4 species: Palmer amaranth, Johnson grass, cocker, and PtxD cotton. Cocker is our wild type, and PtxD, we know that it is able to metabolize the phosphite. These weeds were planted into 25 centimeter by 25 centimeter trays, and we planted them in 3 soil levels, which is 25 ppm of phosphorus, 15 ppm of phosphorus, and 10 ppm of phosphorus. Now the fertilizer dose, since it's a pre-application, it has to go on the ground. We had to apply it as a pre-emergence. So nutrient dose was adjusted based on the soil test recommendation. So what we did was we sent these all 3 soil types for to the soil test for the soil testing, and the results, whatever we got, the recommendations for the phosphorus application, we applied those as our nutrient dose for this pre-emergence experiment. The treatment applications were made with using a backpack sprayer at 3 miles per hour speed and 15-gallon spray volume. The data was collected in terms of emergence count, visual injury, which was basically 0% if there is no injury, and 100%, which means that the plant is completely dead. And biomass was collected at the end of the study. The whole experiment was designed in randomized complete block design with 3 replication, and data were analyzed using ANOVA, and LS means and interactions were considered significant if the p-value was less than 0.05. All treatments in all of the soil types, they were supplemented with other nutrients, for example, nitrogen, except phosphate based on the soil test recommendation. So we tried to provide all other nutrients to the plants except the phosphorus, so that we can see the clear effect of phosphite treatment on the plants. One more thing I would like to notify here, that for my other objectives, as we go down, the pattern, the methodology would pretty much remain the same in terms of the species and the way the treatments were applied. The only thing that's going to change is the dose and the evaluation factors. To begin with the results for the pre-emergence application of phosphite on Palmer amaranth, the first graph here shows the germination percentage. Since it was a pre-application, so germination was counted. The x-axis has our treatments, non-treated, phosphate, and phosphite. And also we can see the clear 3 colors. The gray one is for 10 ppm soil, blue one is for 15 ppm soil, and the dark blue one is for 25 ppm soil. On the y-axis is the germination percentage. So when the phosphite was applied in just to replace the phosphate as a nutrient, we observed that in 10 ppm soil, in 15 ppm soil, and in 25 ppm soil, wherever the phosphite was applied, the germination was less for Palmer amaranth. It was significantly reduced in comparison to the phosphate application and also the non-treated checks. In terms of injury, when the plants grew a little bit bigger, we observed the injury on plants for the stunted height or the plant vigor in general. And we found that in 10 ppm soil, Palmer amaranth incurred about 75% injury, which is a very good control considering it is not a chemical herbicide that we normally apply. For 15 ppm, it's about 65%, and for 25 ppm, it's about 50%. So we see here that as the level of phosphorus, natural level of the phosphorus in the soil increases, the effect of phosphite decreases. So this is one trend that we saw both for the pre-emergence, and I will show you this in the post-emergence study as well. So that means the phosphite interacts with the plants and their growth, and it impacts them significantly. The biomass data, it correlates with the injury. Higher the injury was, the lower the biomass that we collected for the phosphite treatments. For 10 ppm soil, the pictures here show the non-treated, where we did not apply anything, and the phosphite treated plants. We can see there is severe growth reduction. The plants were whitish, or I would say the discoloration effect was there on the plants. 15 ppm, same trend was seen in 15 ppm as well. And for 25 ppm, since there is already enough of phosphorus available, the non-treated plants were also looking good. And the result of the phosphite was seen comparatively okay on the 25 ppm, as you can say that the plants' Palmer amaranth were shorter than the non-treated one. For the Johnson grass, we see the same pattern here as well for the germination percentage. In low P soil, we see more reduction for the germination or less germination. And in high P soil, comparatively, we see more germination. So, but still, these effects, when we compare with the non-treated and the phosphate, were significantly effective for the phosphite application across all soil types. For the injury as well, in 10 ppm soil, we did see about 40-45% of injury on Johnson grass, about 40% in 15 ppm soil, and about 30% in 25 ppm soil. But if we see from our Palmer amaranth injury was high, and the Johnson grass injury is lower comparative to Palmer amaranth. It could be just maybe it's a species to species differentiation, how the plants are metabolizing or using having the effect of phosphite on it. Again, the biomass, it was correlating with our injury data. To show you the impact in 10 ppm soil, these are the plants which were non-treated, not too bad, still okay. But when we look at the phosphite-treated plants, we see severe purple streaking and yellowing in the Johnson grass plants in 10 ppm soil. However, in 15 ppm soil, we did see reduction in growth, but it was more like discoloration. Very less of purple streaking was seen in 15 ppm soil. And in 25 ppm soil, it was pretty much just the growth reduction, nothing else, no other characteristics of low P or P deficiency was observed as we observed in 10 ppm soil. When we look at the coca, which is our wild type cotton, the germination pattern, again, it was kind of similar. Phosphite treatments showed the lower germination, and the phosphate higher germination. It was consistent across all the soil types. Injury, again, I would say here that Palmer and coca both are broadleaf, so they had more impact of phosphite comparative to the Johnson grass, as we see about 70% injury in 10 ppm soil. When we come to PtxD cotton, here we saw that in germination also, the phosphite treated trays had better germination in all soil types, as indicated in these boxes are in all soil types, phosphite has better germination for PtxD. When we looked at the injury data, this is at 14 days after treatment. So this injury data indicates that only 6 or 7% of injury was observed in the low P soil, whereas for other non-transgenic, we saw about 70 to 80%. And only 3% or 4% was observed for 25 ppm soil. And this injury also declined as we were into 21 weeks and 28 days after treatment. Just to show you some pictures here for the effect on cocker and PtxD cotton in 10 ppm soil. When we look at the cocker, this is non-treated, this is treated. We see a bunch of Johnson grass in here, but there is very few Johnson grass and palmer amaranth in these phosphite treated trays. When we look at the PtxD, again, we see the weed scenario. There is less weed pressure in the phosphite treated trays, and the transgenic cotton is doing pretty well. Similar effect was seen for 15 ppm soil, less weed emergence and less rigor growth for the weeds in the phosphite treated trays. In 25 ppm, since there was already enough of phosphorus available, even the non-treated check has plenty of palmer amaranth and Johnson grass available, whereas in the treated trays, we did see a significant reduction in the growth and the emergence of the plants, weeds, I would say here. The next objective is the phosphite application as a post-emergence. As I mentioned, that the species area and the level, how we planted remain the same. The difference is only the doses. So here we had 6 doses. First is the nutrient dose, and second, 2 gallons to the acre of phosphite, 4 gallons, 2 gallons to the acre with 2 sequential applications week apart, and 6 gallons to the acre and a non-treated check. Since it's a post-application, so we did not do the emergence count, but we did visual injury and biomass. To discuss the results here, in the post-emergence, on the x-axis, we have our treatments 1 to 5, and these are the color coding for the soil levels. The peach shade is 10 ppm, yellow is 15, and green is 25 ppm soil. Y represents here the injury on it. So the plants, when we sprayed them, were 3 to 4 leaf stage. And depending on which soil type they were considerably healthy to spray on them and look at the effect of phosphite. So when we sprayed phosphite on palmer amaranth, and in treatments, we can say that treatment number 4 and treatment number 5, they provided almost about, treatment 5 provided 100% control for palmer amaranth, which is 6 gallons to the acre of phosphite applied in low P soil. And in 15 P, which is kind of like medium low soil. So again, the treatment, highest treatment, 6 gallons to the acre, provide about 75% control. However, as we increase the phosphorus levels, the application, since this application, the 6 gallons to the acre was made at whenever the weeds were ready, and evaluations are done 28 days after, we did see that the injury declined, and 28 days after, it had only about 20% of injury. However, in the 25 ppm soil, we observed that when we did the sequential application, because we applied phosphite, say 7 days after, then again at 14 days. So sequential application of phosphite provided better control in relatively higher soil P levels. These are the pictures to show. This is the phosphate treated in 10 ppm. Plants, palmer amaranth is just flourishing there. And with the phosphite, it's all dead. Similarly, in 15 ppm, we see the same effect with the high dose. This is the highest dose applied to the palmer amaranth. 25 ppm, as I mentioned, the injury decline. We did see a little bit of, you can see a little bit of dead plant here. This was pretty much situation when we saw the effect at 7 days and 14 days, but at 20 days, 28 days, the injury declined significantly. Johnson grass, comparatively lower injury levels were seen for Johnson grass. but still treatment 4, which is the sequential application, and treatment 5, which is the highest dose of 6 gallons, provided the best control in low P soils. In 10 ppm soil. In 15, again, it was the highest dose that was applied. Similar trend of the sequential application for 25 ppm, just like I mentioned for Palmer amaranth, was observed in Johnson grass as well. And the biomass data is always correlating with my injury data. These are again the treatments applied at the highest rate, which is 6 gallons to the acre. Phosphate applied Johnson grass, and the phosphite applied Johnson grass in 10 ppm soil at 6 gallons to the acre of phosphite applied. 15 ppm soil, phosphate applied and phosphite applied. And then this 25 ppm soil, phosphate and phosphite. I mean, you could barely tell any differences there. For the cocker, for the post-emergence application, just like Palmer amaranth, we observed the high injury with the treatment 4 and 6. Then again at treatment 5 and again treatment 5 in the 25 ppm soils. For PtxD cotton, for the foliar application, we did not see any injury except for this treatment number 2, which is just 2 gallons to the acre, and 7 days after we observed a 5% injury. For 25 ppm, we observed about 10% injury in 4 gallons to the acre of the treatment, and 2 gallons, it incurred about 10% of the injury. So, but this injury was just at 7 days. It reduced and there was no injury observed after 7 days, like 14 days when I evaluated, there was not any difference between the non-treated and the treated plants. To show here with the treatment number 5, which is our highest dose, cocker, you can see the plant carcasses, not the carcasses, the dead leaves here. And then this is the PtxD cotton, very healthy and beautiful looking. Just to give an overview for this post-application for the weeds, how the weeds were controlled, 10 ppm, 15 ppm, and 25 ppm. The left side, this is all applied with phosphate. And you can see even in low phosphate soils, the weeds are doing fine because we provided them enough nutrition. But when we come into our treatments where we applied the phosphite, we can see the treatment number 4, which is the sequential application. I would say overall, this treatment provided the best control for Palmer amaranth, since it also provided good control in a relatively high P soil. But for treatment, for Johnson grass, I would say among all, it is treatment 5, which provided better control in low P soils, as the effect in 25 P soil was not that much. For the glufosinate and the phosphite study here, the Palmer amaranth and Johnson grass and Liberty link cotton we used, and we had multiple treatments here. The objective of this was to see if the mixing of glufosinate and phosphite can enhance the efficacy of Liberty herbicide. So for that, we used the lower doses of herbicide and had the multiple doses of phosphite added to it. Again, the treatments were applied similarly as I did for the other experiments. To show the effect here, Palmer amaranth at 28 days after in 10 ppm soil. So when you look at this is our treatment where we applied 0.5x rate of Liberty. So it provided 100% control. When we look at treatment number 1, this is 0.25x of Liberty only. It provided about 45% control. So now the treatment number 5, where we applied 0.25x of the Liberty and 4 gallons of phosphite, the injury increased from 45% to almost 85%. So it did provide, it did improve the control of Palmer amaranth. And the biomass data correlates with this. The lower biomass was observed whenever the injury was high, and vice versa. The pictures here shows this is non-treated control where we just had plants not applied with anything, and the treatments, 12 treatments. We can see here treatment 8 and 9, which has the 0.5x rate. And these are the treatment which has 0.25x rate, and treatment number 5, where we had high dose of phosphite, provided good control for Palmer amaranth. For Johnson grass as well, we can say that 0.5x rate provided good control, but in general, since the rates were too low, we did not see much effect of even Liberty on Johnson grass. And if we added the phosphite, as we know from my previous post-emergence application of phosphite, that it is not really very effective on Johnson grass control. So there was not much injury observed on Johnson grass. These are the pictures that shows the effect across all the treatments. Treatment 8 provided good control, and treatment 5, which provided very good control for Palmer amaranth, was not as effective. It did show suppression. There is about like 40 to 50% reduction, but it is not like Palmer amaranth. For Liberty Link cotton, as we know that this cotton does not have the gene to metabolize the phosphite, so the results that you are seeing here are basically because of the phosphite application. So, but where we did not apply any phosphite, there was no injury incurred because it was just the Liberty applied here. For now, the graphs for the slides are just explaining about the effect in different soil types. So whatever I mentioned till now was in 10 ppm soil. Now this is for 15. So similar trend for Palmer amaranth was observed in 15 ppm soil. High injury definitely with the Liberty, but treatment 5 still outstood and provided a good control for Palmer amaranth. For Johnson grass also, we see the similar effect as we saw in 10 ppm soil. For Liberty again, it was just injury because of the phosphite application. Not, I would not attribute this effect to the, of course, not the Liberty. Does not play a role here. The next one here is Palmer amaranth when it was applied to in 25 ppm of the soil. Similar trend with the treatment number 5, where we had 0.25x rate of the phosphite for the glufosinate, and the phosphite application, it increased the efficacy. These are the pictures which clearly shows that the treatment 5 did improve the efficacy. Similarly, for Johnson grass, we did see the same results, and for Liberty Link cotton as well. Just to summarize here, for the soil application, when phosphite was applied, 50 to 80% growth reduction for Palmer amaranth, and up to 40% growth reduction for Johnson grass in low P soil types, compared to the moderate level, which is 25 ppm of the soil. For foliar application, where phosphite was applied alone, the high injury was observed in broadleaf weeds and in the low P soils in all of the post treatments, but there was no significant effect on the narrowleaf weeds. That's what we observed. As I mentioned, that for Palmer amaranth across all soil types, sequential application of phosphite provided better control. In tank mix with glufosinate, tank mix phosphite application provided a synergistic effect for, at least I can confidently say, for the Palmer amaranth, and especially with the 4-gallon application of phosphite, it enhanced the activity. But for Johnson grass, I think we need to explore a little bit more on it. To conclude, yes, this technology is promising. It provides the weed suppression, especially when the level of phosphorus in the soil is less, which means less than 25 ppm of phosphorus. And for foliar application, it is effective in low P soils. And just to mention here that the rates that were used on the higher side, just to see the scope and the level up to which the PtxD cotton can tolerate the highest dose, it was up to 64x of the first 1x rows, or you can say the nutrient dose, and the synergistic effect of herbicide tank mix involving glufosinate for broadleaf weeds. We need to test more herbicides. We tested with glyphosate, it had antagonistic effect. So we are curious to know how this phosphite would interact and provide what kind of results with other herbicides. We need more field evaluations under different environmental conditions, and the conversion of phosphite into phosphate under normal environmental conditions. How does that impact the growth of or weed suppression? At last, I would like to thank my committee members, Dr. Muthu, Dr. Kirti Rathod, Dr. Thomas Isaacate, and Dr. Benjamin McKnight. And definitely a special thanks to Dr. Devendra Pandya, who works with Dr. Rathod, and he was the one who kind of initiated all these studies, and I'm moving forward with it. Daniel Hathcote, he's a program manager in Dr. Muthu's program, and he provides all sorts of help whenever and however it's needed. Last but not the least, Cotton Incorporated for funding this project, and this amazing group of people who always helped me whenever I needed any help. So with this, I would like to thank you all for being here.